Hello friends, how are you? Hope all are fine. Welcome to Sam Biology class. Friends, today we shall discuss about the basic techniques of chromatography. There are two basic techniques of chromatography such as plane chromatography and column chromatography. In plane chromatography, as the name indicates, the stationary phase is coated onto a plane surface, whereas in column chromatography, the stationary phase is packed into a column which is usually made up of glass or metal. The technique of plane chromatography include paper chromatography and thin layer chromatography. Paper chromatography is a simple technique in which a paper sheet, which is composed of highly purified cellulose, is used to support the stationary phase. The stationary phase is water, whereas the mobile phase is a mixture of non-polar organic solvent. The cellulose fibers present in the paper sheet hold water molecules as a immobilized layer due to the formation of hydrogen bonds. This immobilized layer of water molecule act as the stationary phase on which move the mobile phase. In paper chromatography, the sample is applied as a spot on the chromatography paper by using devices such as platinum loop, micropipette or syringe. This figure shows the experimental setup of paper chromatography. This is the chromatography chamber, this is the lead which cover it, and this is a clip with the help of which this paper has been held. And at the base of the chromatography chamber, this is the solvent. And in this figure, the solvent move in upward direction. This type of method is called as ascending paper chromatography, whereas in descending paper chromatography, the solvent is allowed to move in downward direction. In paper chromatography, the components of a sample are separated on the basis of their distribution coefficients between the stationary and mobile phase. This technique is very useful for separation of low molecular weight compounds. It is quick and simple. Another advantage is that this technique offers the two-dimensional operation. Paper chromatography is now primarily used as a teaching tool, having been replaced in laboratories by other techniques such as thin layer chromatography. In thin layer chromatography, commonly known as TLC, a suitable stationary phase is applied as a thin uniform layer on a glass, plastic or metal foil plate by using a spreader. This technique can be performed on the basis of different principles such as adsorption, partition and gel filtration. In this technique also, sample is applied with the help of various devices such as platinum loop, micropipette and syringe. In this technique, large number of samples can be studied simultaneously. It is also quick and simple. Both paper chromatography and thin layer chromatography are based on same principles. The second technique is column chromatography, in which the stationary phase is packed into glass or metal column. After packing, the sample mixture is applied on the top of the column, and after that, the mobile phase, which is commonly known as element, is allowed to move through the column by a pumping system or by applied gas pressure. In this technique, the stationary phase is either coated onto small discrete particles that constitute the matrix or to the inside wall of the column. Commonly used glass columns have a sintered glass disc at the bottom to support the stationary phase. Certain columns are also provided with a suitable support under a replaceable nylon mesh. A cheaper alternative is to use a plug of glass wool with a small amount of quartz sand or glass beads. In column chromatography, during the movement of mobile phase through the column, the sample components are separated on the basis of their partition coefficient between two immiscible phases. Column chromatography can be performed on the basis of different principles such as adsorption, partition, ion exchange and gel filtration. So friends, this is about the basic techniques of chromatography. For more updates, please subscribe my YouTube channel Sam Biology Class, and also please share your comments. Thanks for watching.